Hi, I'm Mrs. Sloan, and this is a video for my AP Biology students. This is chapter 18 in our book currently. I don't know what it'll be by the time we watch this, but this is about the origin and history of life. As always, down in the descriptors of this video, I have the link to what my students use to record their notes. So this is from the College Board, the unit at a glance, the different topics. Of course, this is Unit 7, Natural Selection, but we're going to focus on, in this chapter, in the whole chapter, 7.6, Evidence of Evolution, 7.11 is extinction, and then primarily in this video, the origin of life on Earth. And I encourage you to look through what the College Board, if you check on my website, I have these unit hyperdocs, and it lists off the specific objectives that you need to know and the expectations. So for this video, it's about the origins of life on Earth, and so you can read through that on your own time. And then here's evidence of evolution and about extinction as well. So conditions on early Earth made the origin of life possible. That's the long and the short of it is that the conditions on early Earth, which was basically free, oxygen free, which is a very reducing environment, and it's conducive to building. And so you have these inorganic molecules, maybe outgassings of volcanoes, like what you can see right here, and that the energy on early Earth, that you could take those inorganic molecules and form simple organic molecules. And then with that same energy you could build polymers from these monomers, and then you would use those polymers to build the structures of your early cell, a protocell, and then a self-replicating cell. So that's what we're going to be talking about in this video. So the Earth itself, if you look at the Earth, about 4.5 billion years old, and it took about 1 billion years before you had your first life form. And most of that life and most of the Earth's history, that's been microscopic, relatively recent, that you have multicellularity and the whole skew of organisms that we study in this class. And that last universal common ancestor, Luca, that's the first thing on your notes. 18.1, origin of life, Luca, last universal common ancestor. And early Earth consisted of inorganic molecules, similar to what came out, like I said, of volcanoes. And what we're going to do is we're going to look through these stages. So first you have your chemical evolution. And so let me get out of the way of myself here. So we need to form monomers, then polymers, and then build your protocells. And then we'll talk about biological evolution, how you go from being single-celled prokaryote to eukaryotes, transition from heterotrophs to autotrophs, and then multicellularity. All right. On your notes, you can see those stages. If you just glance through your notes really quick before we get started, you can see stage 1 is evolution of monomers, stage 2, evolution of polymers, evolution of protocells, and then a self-replication system. And then video 2 for this chapter is when I'll talk about the history of life in the major eras and what evolved when, and then we'll look at geologic factors that influence evolution today. Okay, so there's your big picture. So this is a little graphic that I share with my students. I don't know if you can see the stairs here in the background, and inorganic to organic, polymers, and then to cells. Chemical evolution is the first 3 steps up the stairs, and then biological evolution is everything from there forward. So protocells to cells. Prokaryotes that were initially heterotrophs, because it would be easier, make this make sense, you're not memorizing it, it would be easier to consume nutrients available than to use energy from light like autotrophs do to synthesize, take inorganic molecules and make organic molecules through the process of chemo or photosynthesis. So heterotrophs probably came before autotrophs. Along with photoautotrophs, the oxygen revolution about 2.3 billion years ago is when autotrophs were able to split water to release oxygen gas. And then once you have this prevalence of oxygen gas, and at first this was a toxin because everything that evolved in a system of no free oxygen. So when you flood the earth with oxygen, this was considered a poison and killed off most life. And then you had a few that could handle the oxygen and actually use it to their advantage and exploit that and do aerobic respiration, which generates a whole lot more ATP. The oxygen also would create a barrier with the ozone layer so that organisms could move out of the water and onto land. And then you have these multicellular organisms because you have so much more ATP. So that's what the oxygen revolution would bring. I try to remember in billions of years, 4, 3, 2, 1. So you have the Earth about 4.6 billion years ago. And then the 3, for 3 billion years ago, you have your first cells. And then in the 2s, you have the 2 billion years ago, about, within that, you have the oxygen revolution and eukaryotes. And then about 1 billion years ago, you start to have multicellular organisms. All right. And early Earth consisted of inorganic molecules. That's your number 2 on your notes. But living organisms consist of biomolecules that are organic. And so we'll discuss the 4 stages hypothesized for the life to start. Stages 1 through 3, like you see here on the notes. Stages 1 through 3 is the chemical evolution, monomers to polymers to protocells. And then stage 4 is biological evolution. Abiogenesis. So what does that mean? It doesn't have any life, and yet you're synthesizing life. Abiogenesis, the origin of life from non-living matter. All right. See if I can move this along here. So early Earth, probably outgassing from volcanoes, maybe some comet impacts bringing things. So you've got plate tectonics shifting and releasing gases, right, at those divergent boundaries. And at first, water could only exist as a vapor. And then as the Earth cooled, you actually have water existing because of the distance we are from the sun. Water is able to exist in all 3 stages. All right, so our starting molecules, early Earth was reduced. Remember when we talk about oxidizing versus reducing? So when oxygen is in the air like it is now, like if you scraped your car, that metal would rust. And so it's breaking down molecules. But that early Earth, since you didn't have a lot of free oxygen, was more conducive for us to build molecules. And let's see, on your first set of notes on evolution of monomers, the Operin Haldane hypothesis, which I've got going here, I think. Oh, monomer evolution. Sorry, got lots of little pictures in here. So we need to form the bricks of life. So we're going to talk about increasing complexity of chemicals that led to the first cells. And so here, now, these are the 3 big hypotheses. So the primordial soup, I've got that kind of highlighted here because that's most likely. And you have a hypothesis put forth by Operin, which was later Miller did an experiment to support it, that monomers were built abiotically from inorganic molecules using energy present like lightning and UV radiation and geothermal energy, isotopes breaking down from the Earth's core. All of that energy provided what you needed to change those inorganic molecules to organic. And on your notes, the only thing you need to add in there is built abiotically from inorganic molecules present with the energy present. And then I listed it all out there for you. All right. And then the iron sulfur world is focusing on maybe hydrothermal vents being the key to provide those monomers. And then extraterrestrial origins, we don't mean little green men here, but meteorites seeding the Earth with organic molecules. So let's go through those. So what you're trying to build is, when we think about the 4 important organic molecules, right? See if you can remember this. Carbohydrates are built out of sugars, right? And lipids are either fats or steroids. And fats are made out of glycerol and those fatty acid chains, and steroids are made out of 4 rings. And then you have proteins. What are proteins built out of? Amino acids, good. And nucleic acids like DNA and RNA and ATP, what are they built out of? nucleotides. So what you're trying to do is form those sugars, form those amino acids, so you can build the polymers like proteins. So with the energy available on Earth and with the early gases that it is presumed we had, that could form like this organic soup to make these molecules. So the primordial soup hypothesis, early Earth gases, you have most of this, methane, ammonia, hydrogen, and water were put in a cycling apparatus. This was an experiment done by Miller, and here's Miller's experiment. So he put those gases in an apparatus and he circulated them around. You can see he had electrodes right here. This would be like lightning. This heat right here could be like molten magma. And that within a week, all 22 amino acids were formed in addition to other molecules in just a week's time. And that was Miller's experiment, and that supported what Operin had said. So there we go. Had a picture of him. There's Miller. And so within a week's time or so, you had amino acids, fatty acids, glycerol, and nucleotides. So now we have organic molecules. Another hypothesis, but also in conjunction with what we just talked about, is these deep sea hydrothermal vents that remember, we don't have any organic enzymes, right, to facilitate these reactions, but that the molecules, the iron nickel sulfides found in these hydrothermal vents could help contribute either all the organic molecules, or specifically we would need a source of ammonia, right, for nucleic acids and amino acids. So that nitrogen gas, which is the reservoir where you would have the bulk of your nitrogen, could be converted to ammonia with these catalysts that you find in the hydrothermal vents. So that's to be. Further studies hypothesize that nitrogen containing originated as N2 in the deep ocean thermal vents, becoming converted to ammonia. Right? And then I already gave you number 3, the iron sulfur world hypothesis. We just discussed that. And 4 is the extraterrestrial origins hypothesis. Meteorites could have seeded the Earth with the organic molecules or even cells. So the most likely one, at least people lean into, is the Operin-Haldane hypothesis, supported by Miller's experiment. Okay, so now we've got our organic molecules. So now we've got our bricks, right? So now we want to hook those bricks together to build our polymers. So the question is, who came first, right? The code to build the proteins, or did proteins come first to synthesize the building of the code, the nucleic acids, or did they originate at the same time? So evolution of polymers. I think, yep, I got it right here. So how do you make polymers without the assistance of other polymers? So this is the one most supported, is the RNA First, because RNA can act as code, right? mRNA that we translate into proteins. So it can act as a code, and it can also act enzymatic, like ribozymes or ribosomes, right? And then there's the protein first hypothesis, and then back to that iron sulfur world, that at those hydrothermal vents that the polymers could be formed as well. So number 1, RNA first. RNA could be a substrate and an enzyme, a substrate and an enzyme. And protein, let me keep going. You got this. So, clay, sorry, forgot to mention this. Clay also can act as a catalyst. It can hold heat energy and facilitate reactions. Here you have a shared Nobel Prize in chemistry, and that was for the Seck and Altman for the RNA First hypothesis, and just a review of the structure of RNA, which I know you feel comfortable with. And then Sidney Fox, he put forth the protein first hypothesis, and that when hot proteins cool, that they spontaneously will form polymers. And in fact, they will even form these what's called proteinoid microspheres, these sphere shapes, like an early cell membrane. So that is number 2. So heat from the sun could form proteinoids, small polypeptides, that have some catalytic properties, which could then be placed in water and form these microspheres. And do I have another one here? And then this would lead us to, I think you have everything for number 3 already, the iron sulfur world, iron nickel sulfides, already mentioned that. So now we're going to jump into protocell evolution. So this is, you're not considered a cell until you have a code and you are self-replicating. So at this point, you're just carrying on your metabolic reactions, and you have some sort of barrier. So what's happening inside is different than what's happening outside. So you have an outer membrane, and you need a plasma membrane, a boundary, and the word you're looking for there is required, a boundary required for regulation and maintenance of cellular activities. If lipids are made available to microspheres, they will be easily incorporated with the proteins. And since we know, let's read another review opportunity here, our phospholipid bilayer. Think for a minute, what are the structures of a phospholipid bilayer? Right, so we have our phospholipids and they're amphipathic with our hydrophobic fatty acid tails in the center of the membrane and our hydrophilic heads with the phosphates. Then we have proteins that can be peripheral or integral, that remember all the functions of proteins, right? Channels, carriers, cell recognition, receptors, enzymatic, and junctions. We have in animal cells, we have cholesterol, which help maintain the fluidity of the membrane, both preventing it from falling apart and freezing. And then we have carbohydrate side chains in animal cell membranes, right? So the main structures that we need are those phospholipid bilayer and proteins, and that will happen if you look underneath the plasma membrane. It says if lipids are made available to microspheres, they form a lipid protein membrane. And let's see if I have another picture on that one. Yes, here we go. So here's your protocell membrane, your early cell membrane, and then here is our phospholipid bilayer. Okay, microspheres, and this is what Fox proposed, composed of proteins could have evolved into a protocell. And then quasarfit droplets, this is part of Operan's idea, was these fatty layers, and could be a combination of both, right, in order to form our phospholipid bilayer with those proteins embedded in it. Okay, the very early cells, remember who does cellular respiration? All cells. And probably the very first cells were anaerobic fermenters, just participating in glycolysis. And then, so on your notes, nutrition, anaerobic fermenters, only participating in glycolysis. So that was your first supply of energy, was breaking down that glucose for relatively few amount of ATP. But if you're unicellular, that's all you're going to need. Okay, now to be a true cell, you got to have a code and you have to have a self-replication ability to make more of your cells. So on your RNA First, RNA coded for proteins and then hypothesized that reverse transcriptase formed DNA as a storage location for the genetic code. And we've talked about that a little bit in this class, how since RNA is single-stranded, it'd be kind of vulnerable to getting broken down or getting interfered with small interfering RNA. So if DNA could have evolved as a storage, double-stranded, and for eukaryotes protected inside the nuclear envelope, right? So reverse transcriptase could have achieved that. Protein first with Fox, he would say RNA is more complicated. It's more likely that proteins evolve first to guide the synthesis of nucleic acids. And then another individual, Karen Smith, said RNA and proteins could have formed at the same time with clay acting as a catalyst. And I think I gave you everything for that on your notes. And then the genetic code itself, and by the way, RNA First is what most people lean into. That, here, got a little close up here for the new one. Clay is the catalyst. All right. That as far as who came first, we talk about the central dogma that DNA codes for RNA, which then codes for proteins. And we've already revisited that a little bit already, but maybe RNA came first and then DNA evolved as a storage site. Okay, and the evolution of the genetic code, you want to have probably underwent natural selection, to efficiently store genetic information. And remember, we talk about this as evidence of evolution of the universal language of our genetic code. All right. And so our first fossil is about 3.6 billion years old. So that would be more than, right, a prokaryote and a heterotroph. Later autotrophs and later still eukaryotic cells would have evolved. And then here is just showing you those stages. Once again, if you start here at the bottom, you see all the chemical evolution, early Earth, inorganic chemicals, outgassing of volcanoes possibly, and then the energy there on early Earth, lightning, UV radiation, the sun, molten magma, lack of an ozone layer, that all of that energy you could form small organic molecules from those gases, and that was supported by an experiment from Miller, who did it in about a week's time. And then from those smaller organic molecules, that same energy would cause polymers to form. Once you have those polymers, you could build that early cell, a protocell. And then once it became, you had that barrier and you had a code, which was probably RNA first, and you were self-replicating, then you could be a true cell. And then that could lead to all of the biological evolution. All right, and then one more time, here is that review. I went over it with you as well, and you just want to get comfortable with it. And I encourage you to kind of take a screenshot of this. I'll be smaller. Here we go. Take a screenshot of this and put it in your notes because it is a summary of what we just discussed. And I think, yeah, that's going to take us to the end for this video. And I hope you're having a great day. And don't forget to watch video 2.